Good afternoon, everyone, and welcome to Seminars in Oncology. Today's seminar on oncology is the Harriet Tischler Memorial Lecture. This seminar is funded by the Tischler family. Harriet Tischler was a former Dana-Farber patient who succumbed to metastatic breast cancer in 1990. Her family established this lecture in her memory to invite leading cancer researchers to share their latest findings with peers and to educate future generations of oncologists. And I honestly cannot imagine a better person that fits that bill than Dr. Elaine Martis. It's an honor and a pleasure to introduce her. She's the co-executive director of the Steve and Cindy Rasmussen Institute for Genomic Medicine at Nationwide Children's Hospital and holds the Rasmussen Nationwide Foundation Endowed Chair of Genomic Medicine. She's also the professor of pediatrics at the Ohio State University College of Medicine, educated at the University of Oklahoma with a BS in zoology and a PhD in chemistry and biochemistry. Dr. Martis performed postgraduate work in industry at Biorad Laboratories. From 1993 to 2016, she was a member of the faculty of Washington University School of Medicine. Dr. Martis has authored over 380 articles in peer-reviewed journals and has been listed since 2013 as one of the most highly cited researchers in the world by Thomson Reuters. Dr. Martis was given the Morton K. Schwartz Award from the American Association for Clinical Chemistry in 2016 and the Heath Memorial Award from MD Anderson Cancer Center in 2020. Dr. Martis was the AACR president from 2019 to 2020. She was elected a fellow of the AACR Academy and was elected to membership in the National Academy of Medicine in 2019. Dr. Martis is a pioneer in genomic approaches to personalized medicine. She's published some of the earliest and foundational work on how sequencing can be effectively deployed in the clinics. So please join me in welcoming Dr. Martis to present Precision Medicine in Pediatric CNS Malignancies, Clinical Benefit in New Hypotheses. And one comment before she starts is, please post your questions in the Q&A box and we'll get to them after the lecture. So without further ado, welcome, Dr. Martis.
Thanks so much, Matt, for the really nice introduction and especially the opportunity to speak again in Boston, although of course I'd rather be there visiting all of you all rather than standing here in my office in Columbus, Ohio, but these are the times we live in. Hopefully we'll be getting out of them soon. So it's a real pleasure and honor to be invited to give the Harriet Tischler Memorial Lecture. And today I will talk about our work in precision medicine in pediatric brain cancers, talking more about clinical benefit, but also some of the new hypotheses that emerge from that virtuous cycle of patient-centric research that we've been doing here for the last 5 years. So I want to talk first and just kind of set the stage for the molecular characterization of pediatric cancers, really now moving from conventional pathology and immunohistochemistry stains to full genomics and array-based profiling in the effort to provide more precise diagnoses for our patients. And this really corresponds to an IRB-approved research protocol in cancer and hematologic malignancies that we have opened since January of 2018, providing full genomic profiling and extensive and differential analyses, as I'll talk about today, to really study our patients with rare relapsed or treatment refractory cancers, as well as some patients with rare hematologic diseases. So what we have been using is really outlined on this slide in terms of the different assays. So I'll go through those and just note at the bottom that the assays that I'm talking about today have recently been validated in both CAP/CLIA as well as New York State Department of Health in our IgM clinical testing laboratory. And so in particular, we start first with looking at the genome. Here to get high coverage and sensitivity to detect, we actually are using an exome reagent. If you're not familiar with that term, exome refers to all of the 20,000 or so protein coding genes in the human genome. But with pediatric patients where chromosomal alterations, including focal as well as arm or whole chromosome level amplifications or deletions, actually serve a purpose in many disease settings for prognosis for these patients, we actually have expanded the exome, or enhanced it as we call it, adding not only probes to pull down these protein coding regions of the genome, the 1.5% or so of the genome that encodes proteins, but also tiling probes along each chromosome so that we can accurately represent these copy number altered regions as well, either focal or full chromosome or arm level events, as I mentioned. Here, we're also sequencing tumor and matched normal. So we have the ability to appreciate any germline susceptibility that these patients may have as well. And I'll talk a little bit about that at the end. Coupled with DNA, as we know from the early studies that we did with St. Jude back in the day, we really have to also look at RNA data for these patients, because often there's a paucity of genomic changes, and really many of the drivers are epigenetic, and those epigenetic drivers are read out at the level of RNA-seq or RNA expression. So here we're studying RNA-seq data from the tumor, and as I'll talk about, we do multiple analyses on these data, really getting the most bang for our buck in terms of not just the biology program that's underlying the overexpression or underexpression of specific genes or pathways, but also looking at the tumor immune microenvironment in a variety of ways. And then more recently, in addition to our assays, is the Illumina EPIC 850K methylation array. Here we're coupling the results from the tumor evaluation of DNA on this array for 850,000 sites across the genome with these classifiers that have been produced by machine learning based approaches at the DKFZ in Heidelberg. So as many of you may be aware, for CNS malignancies, these methylation classifications are now part of the WHO 2021 guidelines that really are becoming definitive in the setting of diagnosis and coupled with conventional pathology, and in many ways can give us significant clarity around the diagnosis for an individual patient. Then lastly, and again, the learning from large-scale discovery in pediatric cancers is that driver fusions or gene fusions are often the drivers behind these cancers. And so we also have a targeted assay, a pan-solid assay, if you will, using the ArcherDX approach and bioinformatics to identify these driver fusions in the patients in the clinical setting. One of the commitments that one makes when one does large-scale characterization is large-scale computing. And so back in the day, we built large data centers and spent lots of money and time trying to keep those up and running. And with the current hardware changes, we put that all aside when we moved to nationwide and really bought into cloud computing. We have a HIPAA compliant, well-documented environment in the Amazon Web Services cloud to do the analyses on these assays that I was talking about a minute ago. So along the top path here, you see tumor and normal comparisons using the enhanced whole exome sequencing. As I indicated, about 250-fold coverage depth to get good sensitivity to detect. And then alignment using our rapid alignment algorithm, which is named after Winston Churchill. Just a side note that our director of computational genomics is British, and so this is his homage to Sir Winston. And we use this to basically rapidly align these reads as they stream off of the Illumina sequencers and convert BCL to FASTQ so that we can get the answers faster. And the answers here, of course, from tumor normal exome are germline as well as somatic alterations across the many different types of variants that one can identify using the exome data. Along the bottom, then, the RNA-seq also aligns rapidly using Churchill, goes through a variety of different evaluations, including ingenuity pathway analysis, and some differential expression profiling to identify outliers, to perform single sample gene set enrichment analysis. And as I'll talk about today, using estimate analysis and tumor microenvironment deconvolution, we can also study these patients' tumor immune infiltration. This just is an example from our protocol. This was just updated as of the end of February this year for CNS histologic diagnoses that have been studied. So you can see the numbers here in the pie graph, and then across the bottom of this x-axis, you can see the different gross scale categories, such as astrocytic and oligodendroglial tumors, embryonal tumors, et cetera. So just for reference, about 52% of the tumors that we've studied so far, about 357 in total patients have been enrolled on this study so far with cancers. 52% of those patients are in the CNS malignancies. And so the bottom line has been illustrated beautifully by already published papers from St. Jude, from the DKFC, and from the Australian Zero Childhood Cancer is this: comprehensive profiling yields answers for these patients. These are examples from our patient population. As I mentioned, 350 patients, 357 patients studied so far. A little north of 400 are enrolled and in the process, the difference are in the process of being studied now, where we can report out aspects of germline cancer predisposition, somatic fusions, potentially targetable gene expression, medically meaningful copy number variants, and then medically meaningful somatic variation. And the bottom line is that for each of these patients studied, over 95% had at least one medically meaningful result. Some 10% or so have actually changed diagnosis. And in many ways, of course, as we know, changing diagnosis changed the treatment plan in many cases for these children. So it's an important component of determining how they will be cared for, and ultimately what their outcomes will be. And so we're now starting to evaluate outcomes, and we're also trying to metric the provider's utilization of the data to change diagnosis or treatment. And I think this is an important coda on the description of this protocol, which is that we really found that the more we became involved in profiling these children's cancers, returning the results during different molecular tumor boards for neuro-oncology, for sarcomas, for rare cancers, and so on, the more our providers started reaching out to us when the challenging patients were coming into clinic, and really began to embrace and understand what this type of comprehensive profiling can provide to them in the care of these children. So it's been really exciting to be a part of that. I want to focus now on brain cancer, and a lot of my own personal research that I'll talk about now really focuses on brain cancer for the precise reason that's listed here. It now has surpassed leukemia as the predominant cause of death by disease in children and death by all disease. And so really this is a pretty intractable problem, just like in the adult glioblastoma setting. We're also doing a little bit of research there that I'll mention along the way. But I think this is remains a hard target, as we like to say. And in particular, what I'll talk about is really exploiting this CNS cancer immunity connection, as illustrated in this beautiful review by Ira Melman in 2013 in Immunity, really talking about the fact that the CNS has its own lymphatic system. This was first described in the mid-2010s, and really that the historical thinking about brain tolerance and the immune privileged environment of the brain needs to be revisited. In particular, of course, the reason that we're interested in the immune microenvironment in pediatric cancers is because, as Matt and I were talking about before we started today, immunotherapy is really one of the most promising therapeutic classes in cancer medicine today. And we've moved well beyond immune checkpoint inhibitors to a multitude of different therapies. I'll focus a little bit today on personalized vaccines and oncolytic virotherapies. But I think in the pediatric setting, it's also a bit specialized because we do understand and appreciate that the pediatric immune system is quite a bit different than the adult immune system. And some of these relatively blunt hammers like immune checkpoint may not work so well in children, and we may need to tailor some of our therapeutic approaches to their specifics of their immune system. Of course, in all of this, preclinical as well as clinical trials, we've got to have platforms and data analytics to really tease out the elements from the correlative science that we're trying to do that tell us the right way to move for these patients, whether they're animal or human, to really perfect these approaches. And so what we've been doing, and I'll talk a little bit about today, is really developing these platforms and data analytics, starting with the comprehensive DNA RNA profiling that I talked about a minute ago, but also really now coming down to things like single cell and digital spatial profiling, which I'll also talk about as well, and really beginning to study what happens over a therapeutic interval for any one of these types of immune therapies or other therapies that we might like to try. So just a few brief comments on some of the tools. I'm sure many of you are very familiar with these, so I won't spend a lot of time on them. But obviously, one of the approaches that has come our way, and there are many, including Shirley Liu's timer, as well as Excel and others, is imputing the tumor immune microenvironment. So taking that RNA-seq data from the tumor and really starting to look for signatures of immune cells of different types and trying to figure out their relative proportions. And as I mentioned a minute ago, whether those proportions and activities change over time. So here's just an example, which is CyberSort and CyberSort X, one of the several imputation algorithms that we've been using in our work. One of the other truths, of course, in the clinical setting is we can't always count on nice, fresh, frozen material from our tumors to give us nice, high-quality DNA and RNA, and this is a constant struggle. I'm happy to say that many of the neuropathologists that we work very closely with have recognized this and now routinely set aside nice, fresh, frozen sections for us to do our thing in genomics, but of course, with historic samples, such as the longitudinal ones that I'm talking about today, we can't always count on that. And so one of the alternatives to the imputation that I just talked about is the use of this nanostring platform. Again, I think many of you are familiar with the barcode-based approach of nanostring that can detect about 850 different species according to multiplex barcoding using a counting approach as shown on the right-hand bottom side of this slide. And here, again, we're just using an immunoprofiler assay, so sort of a non-customized, commercially available platform to really understand immune response categories, so different types of cells, and in some ways the signaling that's going on in those cells. And I'll show some examples of this with a specific patient that I'll talk about. Then the work that we've done in neoantigen prediction also turns out to be in some cases an exciting angle to pursue. I won't talk about this extensively today, but to just give a shout out to my former graduate student, Jasreet Hundal. I was happy to work with her on both of the manuscripts and the work that went into them at the bottom of the slide. And most recently in cancer immunology research, a couple of years ago now, we published the PVAC tools suite, which is shown here, a figure from the paper. I'm really showing how with the combination of DNA and RNA data from exome and RNA-seq, we can compute things like HLA type, even look at specific fusion peptides as potential neoantigens using the PVAC fuse pipeline, and having a really augmented, perfected, high-quality PVAC-seq pipeline that improves upon the one that we initially reported in 2016. We've even gone so far as to use this platform to predict neoantigens in a variety of clinical trials, one of which we published a few years ago in Science on dendritic cell vaccines for melanoma, and a study that we have yet to publish that is still ongoing in glioblastoma in collaboration with Gavin Dunn, who used to be at WashU and now is back at Harvard. So just another one of the tool sets that we have put together and offer as usual as open source code at the pvactools.org source and GitHub. Okay, so speaking of Gavin Dunn and his laboratory, I wanted to just take a quick segue into what we've done in adult GBMs, comparing those to a variety of brain metastasis samples from patients. And this was published recently in Cancer Discovery electronically in 2021, and the full version came out early 22. So I'll just show you briefly what we've done here. You can see the sample set described on the this figure here, 15 primary gliomas, 4 recurrent gliomas, and 11 brain metastases. And what we were able to do through Gavin's careful work in the operating room was to take different sections from these brain metastases as they were resected and perform multi-sector tumor sequencing. So 92 tumor regions in total from these 21 patients were studied. And then we also, sorry, it's more than 21 patients. 21 were treatment naive. There were recurrent GBMs with standard of care therapy and 5 brain metastasis patients. So we had no prior immunotherapy for these patients. And as I mentioned, 92 total. So 2 to 4 spatially distinct tumor regions were taken from each patient. And these were some of the assays that were performed, as I mentioned. And so what did we find? Well, first of all, we're sort of wanting to compare heterogeneity for gliomas versus brain metastases using this multi-region sequencing. We found that a median number of aggregate somatic variants per tumor were higher in brain mets than in either primary or recurrent gliomas, so an overall higher mutation load. But then when we start to look at the intratumoral heterogeneity going through a neoantigen pipeline, as I just described for PVAC-seq, we found that in terms of identifying neoantigens, as if one were going to create a personalized vaccine, for example, in brain metastases, we hit a limit of sampling where we began to find more variants, unlike in primary and recurrent gliomas. And when we turn that into neoantigen prediction, we actually found that with brain metastases for class 1 and class 2, neoantigens were more numerous, more clonal, and more likely to be captured by a single site sampling than were gliomas. So again, a nod to the heterogeneity of this high-grade gliomas. And then finally, in terms of immune cell infiltration, we took a look here at brain mets versus gliomas for PD-L1 as well as CXCL4 levels. And these were reflective of what's already known, which is severely immune suppressed tumor microenvironments in gliomas compared to brain metastases. And finally, by further characterizing the cybersort-based macrophage infiltration, we showed a slight polarization towards immune suppressive M2 phenotype in gliomas compared to brain metastases, whereas brain metastases skewed toward higher expression of monocyte-derived macrophages relative to microglial specific enrichment in the gliomas. And so some substantive differences here that tell us a lot about the differences in immune cell infiltration and that immune suppressive environment that they turn up. And so the reason for sort of going through this is to point out the study, but also now to dive into the contrast that we find in the pediatric setting to what's there in the adult setting. So I want to spend just a minute to talk through a case study. This will illustrate some of the tools that I've talked about, and I think we'll bring in some of the differences and set the stage for the rest of the talk. So we're looking here at a patient that was 10-year-old male presenting with a recurrent CNS cancer in 2017 when he came to our attention, a so-called high-grade neuroepithelial tumor. And we contrast this with his 2015 tumor that was gross totally resected, that was called a mixed glioneuronal tumor, neither of which are terribly precise. In fact, David Ellison at St. Jude referred to the 2017 tumor as anaplastic ganglioglioma. One tumor was formalin fixed, paraffin embedded, one was fresh frozen. And in the process of doing our tumor exome sequencing and RNA-seq, the RNA-seq analysis came up with this fusion with EP300 and B-Core. Now, many of you may have heard of this fusion. In fact, you'll know why in just a second. Many of you may have heard of it, but at the time we studied this patient in 2017, there was not evidence that we could find in the peer-reviewed literature for this patient in the context of this being a known fusion. So the patient went on to receive additional radiotherapy post-surgery and some standard cytotoxic chemotherapy. In the meantime, a group that includes several folks at Harvard produced this result and publication in April of 2019, now reporting on 3 different cases with EP300 core fusions, as shown here, slightly different in their nature. And in fact, one fusion actually matched our 2017 patient. Importantly, one of the things that came through in this paper was that EGFR was overexpressed in these patients. And indeed, if we look at our patient in terms of the RNA-seq data analysis for expression, you can see in the red bars evidence from the primary disease of high EGFR overexpression, a little contextually lower in the recurrent, but we also noted, as I'll talk about in the next slide, that this patient had a significant difference in the immune infiltration, thereby giving some dilution to the RNA signature because of the amount of immune infiltration. So what does that look like? Well, we looked at it in 2 different ways. Our standard pipeline runs estimate scoring, and you can see here the primary tumor with an immune score that's highly negative, and the recurrent tumor with an immune score that's quite positive by contrast. So additional information supporting the immune infiltration on top of what conventional pathology stains told us. And then here are the CyberSort X analyses, again, from the tumor RNA-seq data. The primary tumor is here on the top analysis and the recurrent tumor on the bottom, and quite striking differences, in particular, according to the M2 macrophage infiltration for this patient, as well as M0 macrophages, M1s, and monocytes. So these additional orange and yellow colored bars next to the large M2 macrophage signature. And so just to work on this a little bit more, we took a look at the comparisons between the tumors in terms of the variants that were identified in the primary tumor here on the left and the recurrent tumor. You can see an uptick in the number of mutations that are identified in the recurrent tumor, and shared a couple of mutations that aren't meaningful in the context of cancer, but are definitely shared between the 2. So this substantiates from a genomic level that the recurrent tumor is indeed recurrent and not a second primary. We also calculated the PVAC-seq neoantigens that could be identified from the variants that were in these 2 tumors, and you can see that with a binding cutoff of 500 nanomolar, which is standard, really just one neoantigenic peptide emerged from that result for the primary, and additional ones that were identified in the recurrent tumor based on that binding cutoff, so from predictions. And then we did want to go back and compare this to some pathology-based immunohistochemistry stains, first for CD45, just a broad hematologic derived marker, but then CD68 for monocytes, 163 for macrophages. And what you can see here is that across these different stains for the recurrent tumor, JL36R, there's a significant uptick in staining compared to the primary tumor that's shown on the bottom panel. So we used a lot of the data that I talked about, not just for this one patient, but for multiple patients that I don't have time to talk about today, looking at these longitudinal aspects of tumor immune microenvironment and showing that in high-grade disease in particular, we do see pretty good evidence in most patients, admittedly a small number that we've been able to collect these longitudinal samples and do the studies on. But really supporting the fact that immune therapy may have a role in these patients because of the immune suppressive environment that gets set up by M2 macrophages, as I mentioned earlier, for glioblastoma. So we partnered with several of our colleagues that are interested in developing new immunotherapies, and here at the hospital, as well as at another hospital in New York, and we're able to get funding under the National Cancer Institute's Cancer Moonshot Pediatric Immunotherapy Drug Discovery Network, or PIDDN. So this is a U54 that got granted right about the time everybody was shutting down for COVID. So we were a little bit delayed getting going on it, but we've made good progress since then. And these are the therapeutic modalities that are being investigated in the different projects. I won't talk about them much other than oncolytic virus-based therapies here listed number 2. But we're also evaluating natural killer cells that can be engineered specifically, as well as targeting myeloid cell-based immunosuppression. And my role, of course, is co-PI, but also conducting the comprehensive immunogenomic profiling that I've been presenting to you. And we're also happy to collaborate up to OSU. Greg Bebahani, who trained in Gary Nolan's lab, is our mass cytometry expert up there. And the single cell work is going on here at Nationwide Children's Hospital. I'll talk about all these things. So first, I want to just mention, lastly, in terms of the tools that we're using, a new platform that we've also been evaluating for immune microenvironment studies, and this is digital spatial profiling using the geomics platform. I think many of you, again, are familiar with this. It's not truly a single cell level evaluation, but rather looking at regions of interest that can engage anywhere from sort of 30 to 50 cells in these regions of interest. We identify the ROIs based on fluorescent antibody staining, a pancytokeratin, and a CD45 to look for immune cells. Once those ROIs are identified, then the instrument itself will use ultraviolet light to cleave off the oligos that label oligo-labeled antibodies. So the fluorescent ones are used to locate the ROIs, but then we have a pool of about 80 additional antibodies that are antibodies specific for cell surface markers on different immune cell types. These cleaved oligos then can be placed in a 96-well plate according to the regions of interest. They're aspirated and then taken over to the nCounter platform for a readout using, again, the barcode-based oligo system that I showed earlier for the immunoprofiler. And then ultimately, we take this information back to the geomics platform and marry together the regions of interest with the actual profile that's been identified for that ROI. So I think the last thing to point out about this platform, as many of you may know, is that there is a new capability that's become available where we can take not the same slide, unfortunately, just yet, but an adjacent FFPE slide and evaluate the transcriptome profiles from the same regions of interest where we've studied the antibody binding. So I think this is an important add-on because we'll obviously get the cell surface marker description of different immune cells that are in the regions of interest, but also we'll be able to appreciate the transcriptome-based activity coming out of those immune cells, including signaling of different types, as well as immune suppression versus not. Just want to show a couple of quick examples from, again, a longitudinal patient study. Here, this patient, CH42, is one of these unique individuals we see in about 50% of CNS malignancies, initially presenting with a medulloblastoma, receiving some radiation-based therapy for that medulloblastoma, as well as surgery and chemotherapy, and coming back with a radiation-induced second primary glioma, which is a much higher grade. And as you can see, has some known driver mutations, does not have the ISO17Q that was present in the medullo, and has an increased macrophage infiltration in this setting. Also studied in this run on the geomics were two other tumors, an xanthoastrocytoma that is a hypermutator, also increased M2 macrophage evidence. And then this very unique patient with a neuroepithelial tumor. This is a patient with Lynch syndrome. As it turns out, as you can see, biallelic PMS2 loss. I would actually call him an ultramutator, probably more than a hypermutator. But interestingly, in the primary setting that we studied here, no evidence of immune infiltration for this patient. But let's focus on CH42 for just a minute. We can see further evidence here, as I illustrated previously, looking at primary tumor mutations and recurrence. There is nothing shared between these two tumors, so the recurrent tumor is truly a second primary. And as I mentioned already, there was a PTPN11 or SHP2 that was unique to the recurrent tumor, so some potential there for druggability, but two distinct genetic profiles. CyberSort comparison for these two samples, the recurrent now shown on the top, the original medulla on the bottom. Sorry, this is not recurrent, it's a second glioma. And I apologize, the color scheme has changed here rather. But again, the dark purple here indicative of significant M2 macrophage infiltration. As I mentioned, also some M0 macrophages are evident as well, although there are some present in the primary tumor as well. So really reminiscent of that patient that we just talked about a few minutes ago in the comparison of primary to recurrent. Now we bring in the geomics of protein counts. So here we have the primary in blue and the recurrent or secondary cancer in green. And these are the two hypermutators, the Lynch syndrome patient on the left and the other patient with the xantheroastrocytoma on the right. And what you can see and appreciate, I think here are upticks significantly in CD56 for both patients, but a significant increase in CD44 cleaved caspase and now granzyme B appearing in the setting of the recurrent tumor. So just a few markers there of increased immune activity. And if we focus specifically on M2 macrophages, you can see the CD163 differences significantly elevated here in the recurrent compared to the primary. So I want to switch gears now just for the end of the talk and tell you about a little bit of the preliminary single cell studies that we've been doing in the preclinical setting now with the U54 funding I mentioned a few minutes ago. Just first to introduce two of the medulloblastoma mouse models we've been using. These are a kind gift from Martine Roussel at St. Jude. And this is an NMIC model and a CMIC model that are sort of described in how they were created here on either a corresponding column. And these are obviously published models. And then, as I mentioned, I was going to talk specifically about our OHSV, oncolytic herpes simplex virus based approaches here. And this is just the engineered virus description, if you will. This is using next generation OHSV, commonly referred to as C134. This is a chimeric virus, and it produces a protein that we can actually look for in the single cell expression data. And I'll show you an example of that, I think, if I still have it in here. We're working here with Kevin Cassidy, whose group shown at the lower right-hand corner has already published on this OHSV. And Kevin also has been part of a clinical trial going on in adult patients with recurrent glioblastoma using C134 in an FDA IND approved trial where the tumor resection bed on these recurrent patients is seeded with the C134 cocktail, and then they are observed in clinic. And I'll talk about that trial in just a minute, but this is not just in the preclinical setting, it only is for our U54 studies. So this is just a paper that was recently published from that clinical trial that I just mentioned. Katie Miller, who's now a research assistant professor at IGM, was the lead author on this. And this is in clinical cancer research, as I mentioned. So these are just a couple of quick figures from the paper looking at recurrent glioblastoma responses to the OHSV treatment. And what you can hopefully appreciate, although this looks a little blurry to me, I don't have my glasses on, so maybe that's why, is we're looking at the comparison between responders and not in black and non-responders in the gray. It's so-called characterized based on their interval of disease-free survival following the surgery and the application of the OHSV cocktail that I mentioned. And we're also looking at pre-treatment versus post-treatment. So pre-treatment is the orangey yellow and post-treatment is the green for these different patients. And really looking here using the immunoprofiler assay that I mentioned at a variety of different immune cell infiltrates. to really study the sort of pre-post interval. So if we look carefully at pre versus post in this responder, which is sort of the central column here, you can see a very different profile compared to some of the other pre-post for the responders versus not responders. And we broke that out into a variety of different box and whiskers plots shown down here for total tumor infiltrating lymphocytes. T cells, we can see, is quite differentially involved depending upon whether you're a responder or not a responder. And the code is over here. Dendritic cell infiltration and natural killer cells, for example, are all elevated in our responders that are essentially reflecting the way that the oncolytic herpes virus treatment is bringing in components of the immune system based on this treatment in patients that are avid responders. And then we just perform this difference in the mean-centered cell type scores from pre to post-treatment, illustrating that much of what's happening in the post-treatment setting for responders is due to this differential immune cell infiltration. So if we go back to the preclinical model now for just a minute where we've done our single cell work, we know that just from gross studies in terms of overall survival comparing vehicle to C134 treatment for different mouse models. So I talked about the CMYC model here and the MYC-N model, which is a sonic hedgehog model. We can see that C134 does increase the median survival in both of these mouse models, as well as another form of the patch-associated sonic hedgehog model. If we take the RNA-seq from these C134 treated mice compared to vehicle-treated mice, and we impute the immune changes following OHSV, so this is sacrificing the mice at typically 5 to 6 days after the use of OHSV treatment, what we can appreciate here in these top bars is really significant M1 macrophage and probably transition from M0 in the vehicle treated to M1 in the C134 treated mice, as well as an increase in most cases of CD8 cytotoxic cells. We then produced some of these immunoprofiler volcano plots comparing day 2 to day 6, and really just trying to illustrate here, this is just for the CMYC model at day 2, comparing C134 to vehicle, that in the differential for C134 treated, we are seeing an uptick in a number of specific immune markers. And then at day 6, a very different set of immune markers that emerges. And so this is sort of the just post-treatment and then followed by the day 6 animal being sacked and evaluated on the immunoprofiler. So we're seeing some sort of changes over time, although it is admittedly a small time interval relative to the patients that were being studied in that clinical trial. And what we've done next is then taken these same mouse models using the either virus or vehicle injection. And once we've sacked the mice, we then collect the tissue, perform single cell RNA-seq for the brain tumor, as well as much of the brain, because the brain tumors are a little hard to separate out. So what we're seeing here is just sort of the medulloblastoma based UMAP projection for mostly the tumor cells here in this large purple blob. And then, you know, my MSTP student, Jack, has been working hard to try and make these cluster assignments for these immune cell clusters shown here. And you can sort of see hopefully see a blow up where he's more carefully delineating the immune subset analysis away from these gross descriptors to things that are a little bit more indicative of what's going on here, including this large blue cluster, which corresponds to M2 tumor infiltrating macrophages, or tumor-associated macrophages rather. And some of the ways that these assignments are being done are sort of illustrated in this plot Here on the lower left, where we're really looking at the average expression and then correlating that to known expression profiles, obviously, for these different types of immune cells in order to make the cluster-based assignments. So it's a little early days, but we think we're able to show these differential changes in terms of vehicle treated versus oncolytic herpes virus treated, in terms of eliciting in immune subsets. And so we plan to continue to pursue this in a variety of different ways. And then just to finish up, you can see here represented data for day 2 vehicle versus day 2 C134. Here I'm looking at the x-axis, day 6 versus vehicle versus day 6 oncolytic virus for the CMYC model on the left and the MYC-N model on the right, where we are seeing significant changes in terms of the immune infiltration on these day 2 virus-treated samples, according to the different assignments over here for microglia and macrophages most prominently, which largely correlates with the single cell data that I just showed you a minute ago. So I just want to finish up by concluding that, we're lucky to be able to study these patient samples, and I think start to formulate and explore hypotheses, much like I've talked about today. We do have multiple lines of evidence for macrophages and monocytes in the TME that are recruited from the periphery. We know that when we study the primary cancer, and we're lucky enough to have it and to be able to study it with a variety of different approaches, as I've talked about, that these are minimally present in that primary setting, but enhanced in the recurrent setting. And these likely provide, as I mentioned, an immune suppressive context. The presence of neoantigen coding alterations may present an avenue to engage the immune system. And here we're really thinking hard about how to identify neoantigens in our preclinical models and perhaps start to engineer those oncolytic herpes viruses to not only bring in, generally speaking, immune system components, but maybe some that are specific for neoantigens and therefore even more selective. And we know that we need to pay attention, as I mentioned in the beginning, to differences in the pediatric immune system and really try to come up with therapies that are going to be appropriate for children being treated for brain cancers. And very happy to be part of this U54, as well as the network that goes along with it, which is very active and collaboratory. Then just one other thing to mention, if you'll give me a little bit, just a minute or two here. I did just want to make one sort of big announcement that will be officially announced in a press release from the National Cancer Institute on Monday of next week. And this is a very exciting offshoot of the work that we've been doing with children in our hospital. We're really fortunate to be now working with the Children's Oncology Group initially to begin changing the way that children in the United States are characterized, who develop cancer, are characterized with these multiplex analyses that I've talked about. And so the NCI is going to be providing reimbursement to our laboratory to perform clinical genomic testing of pediatric solid tissue malignancies for the various clinical trials groups that the NCI sponsors, again, starting initially with COG, but maybe opportunities to bring in the Pediatric Early Phase Network, PEPCTN, PBTC, which focuses exclusively on brain cancers, and possibly others that are exploring immunotherapies. This will be clinical testing using tumor normal exome, the Archer fusion plex, and the methylation arrays. And importantly, we think we're going to be able to get these samples in the door, get the analytes over from our biopathology center here at Nationwide, and perform assays and sign out and return results in a 14-day turnaround time. That's a pretty Herculean effort, but what we've put in place are a lot of electronic approaches that replace often sheafs of paper that come over with every patient sample, hopefully streamlining the accessioning of these samples. And then obviously, coming up with a variety of large data sets that will ease the really challenging part of all of this, which is turning around not only the assays, but the analysis and interpretation of those assays over time. And I don't have time to talk about that today, but hope to come back in the future and tell you about some of the systems that we're putting together for making that more rapid and more scalable, because that's really what it's going to take to get to 3,000 cases a month, which is ultimately the goal here. Finally, we're using our secure Amazon Web Services portal to actually provide out clinically signed out PHI containing results to these providers in a secure way, and to perform that again electronically, so that they're redacted from PHI in cases where they need to be for the purposes of record keeping, containing PHI when they're going back to that clinician that enrolled that patient onto the COG trial, et cetera. Finally, the last thing that I think is really critically important here to mention is the last bullet point. We're not going to be sort of putting these data away for the ultimate publication of something, that's a clinical trial. We're actually going to be actively depositing these data into the Childhood Cancer Database Initiative public data repository within 90 days of the sign out of these test results. So part of this system over here on the right is actually automated transmission of these data into the CCDI, along with PHI redacted clinical data that's really going to enhance the data mining environment for pediatric cancer moving forward. And I think that's probably the purest notion that really got us going and interested in participating in this. So I'll finish and really just acknowledge the team. It's gotten quite large, and so I have some sort of gross descriptors here of the different services groups. But it does take a village to make this all happen, and especially want to acknowledge all of the physicians that have collaborated with us in the development of this protocol. And as always, special thanks to our patients and families. And finally, just want to acknowledge the Mark Foundation for Cancer Research, who's funded Katie Miller's research for a long time, and much of what I talked about today in the immune microenvironment is work from Katie. So thanks very much, and I'm happy to answer any questions that might have come up during the talk.
Thanks so much, Elena. That was fantastic, and the scope is incredible, and I feel really in paradigmatic of where the field is going.
I hope so.
and really to your sort of last slide, if you envision this sort of being this service being available, like sort of nationwide or for even adult cancer care, like do you envision sort of like multiple centers like across the country that do this, or what's your sense for sort of even scaling beyond the samples that you're talking about now?
Yeah, thanks, Matt. I love that question. I do, I really hope this is a 5-year pilot that just illustrates the importance of this, multiplex characterization. Obviously, we're riffing a bit off of work that's already been done, as I mentioned, and published. But one of the things that really came through from the DKFC study from Inform, in particular from that cancer discovery paper, is we really have to start moving this characterization earlier in the cancer journey rather than waiting. And so I think that really speaks to your question in terms of the need and how we hope to illustrate that over the next 5 years. I think the scalability question is a really important one, because at the end of the day, I know that there are not enough clinical directors and molecular pathologists, at least not here, to really scale to the level that we would need for cancer diagnoses across the United States. And I especially worry about access to this, right? So any kid at any even community hospital, like what hope do they have of actually getting access to this? So I think if we're going to really be successful at that important job, we are going to have to come up with distribution systems where, much like the Amazon Web Services clinical report out, sort of communication that I mentioned, instead, what we're probably going to need to do is to move the data that we have from these primary assays into a cloud-based environment compute on it as much as we can, and then have a distributed system of molecular pathologists and clinical directors that can be engaged in the analysis of the interpreted data and the sign out. So I think it's two things. I think it's really building these informatics systems and machine learning based approaches that can combine the fusion tumor normal results and the methylation when needed to a more of a comprehensive sign out, if you will, because that's really what we use the data for in aggregate. But it's also going to require a distributed network, I think, of these specialists to interpret the data fully and sign out the clinical reports.
Thanks. And again, if folks have questions, feel free to place them in the Q&A box. But I actually have another question too. Like in terms of like data types, you know, you had mentioned like RNA, DNA, methylation. Do you think that one of those data types tends to predominate or is more important? Do you think that this is the only data types that we're going to need? Do you envision this sort of like growing out into like, you know, other like metabolites or, you know, other things that we can conceive? Is this something that's going to be organic and sort of grow as we learn, or do we know all we need to know?
I don't want to rule anything out, but Matt, as you and I were talking about before, often what people focus on is sort of, well, how much does all this testing cost? And, you know, is it really, you know, necessary? And I think, you know, one of the areas I'm pretty passionate about is getting past that and sort of looking at the impact of this testing in a more of sort of the environment of health economics that surrounds patients with a cancer diagnosis. And no more important than in the pediatric setting, of course, because we know from long-term studies that these patients, if they're lucky enough to survive their cancer, based on current modalities of cytotoxic chemotherapy, high-dose corticosteroids, and often radiation therapy, we set these patients up for a lifelong journey of poor health and a high, you know, rate of secondary cancers. Let's just be honest about that. And so I think people tend to not think about how much that costs as a lifelong health burden associated cost, quality life years, adjusted life years, if you want to talk in insurance parlance. And ultimately we're going to have to, but you know, but what would change about that, and would we stop wringing our hands about the cost of molecular testing and start thinking about, wow, if we could do this earlier, it's actually a minimal cost compared to the cost of maybe identifying a very effective targeted therapy earlier in the cancer journey that, because of its lower side effect profile, sets this patient up for less of a, you know, a long-term health, you know, sequela scenario. And so I think that's more kind of the lens that we need to start putting on it. And, you know, I've started talking with hospital leadership about how we can engage our health economists to really start quantifying what is one thing versus another, and how, you know, can we push towards that more ideal total cost of care over the lifespan, rather than really focusing in on things like how much does molecular profiling cost. Maybe that's self-centered and overly hopeful, but that's kind of the way I think we want to try and spin it and see where we get to.
Yeah, no, that makes a lot of sense.
I hope so.
So yeah, I guess if there are no other questions, I just really appreciate your time and just the breadth of what you're doing, and appreciate what you're doing, and also to the Tischler family for supporting these seminars. So thanks so much, Dr. Martis.
Yeah, thanks again for the invitation. Really appreciate it.
So.